Okay, Assalamualaikum and good afternoon everyone. So today we are going to continue our class with the topic for this week is the sensory system. So we are going to continue about the mechanoreceptor, photoreceptor, magnetoreceptor, and also the last types of sensory receptor, which is the thermoreceptor. So before that, let me share my slide. Okay, hopefully all of you can see my slide. Okay, as you still remember from the previous class, we have the classifications of sensory receptor, which is the first one is based on the stimulus locations. Okay, we have the telereceptor, which detect the distant stimuli. We also have, for example, of the distant stimuli like visions and hearing. We also have the exteroreceptor, which detect the stimuli on the outside of the body, for example, pressure and temperature, which also have the interoreceptor. Okay, the other classification of sensory receptor is that based on the type of stimuli. So we have 5 here: chemoreceptor, mechanoreceptor, photoreceptors, magnetororeceptors, and thermoreceptor. So for today, we are going to continue about these mechano, photo, and magnetoro, and also the last one is the thermoreceptor. So for the mechanoreceptor, okay, so it will transform the mechanical stimuli into the electrical signals. Okay, so mechanoreceptor is regarding the transformations of the mechanical stimuli into electrical signals. So all organisms and cells can sense and respond to the mechanical stimuli. So we have two main types of ion channels that involved in this transformations of from the mechanical stimuli. So we have the INEC and also we have the TRP. So the INEC is the epithelial sodium channels. Okay, and the second ion channels that involved in the process is the transition receptor potentials or TRP. So for example, in here we have the examples of INEC channels and also TRP channels. So for example, in here, in the C. elegans, the nematode, okay. So this is the mechanosensory neuron, okay. So in here, okay, so they have the INEC channels, okay, and also in the flies, okay, the TRP channel. So they have the sensilla, okay, and also they have this is the cuticle and this is the mechanosensory neuron. So in here they have the TRP channels, okay. So there are three classes in the touch and pressure. We have the first one is the baroreceptor, the second one we have tactile receptor, and the third one we have proprioceptors. So the baroreceptor is actually the interoceptors that detect the pressure changes. Okay, the interoceptors, the locations, okay, the stimulus locations, okay, which is the interoceptors. And the second classes of touch and pressure is the tactile receptor, which is the exteroceptors that detect touch, pressure, and vibrations on the body surface. Okay, the tactile receptor is regarding the correlativity touch. And the third one we have the proprioceptors. So this is functioning to monitor the positions of the body. So we also have two types of mechanoreceptors in insects. Okay, so we have type 1 cells and also type 2 cells. So the cellular structure, both for the type 1 cells, they have ciliated bipolar neurons, And for the type 2 cells, they have non-ciliated multipolar neurons. The locations, the type 1 locations is located associated with cuticles, while the type 2 cells, they are associated with muscles or connective tissue. And the ion channels that involved in the type 1 cells is a TRP, and the type 2 cells is the NAC, okay? And the function of type 1 cells is for proprioceptions, which is to monitor the positions of the body, and also for touch and hearings. And for type 2 cells, it's functioning in a stretch receptor and also proprioceptions, which is monitor the positions of the body. So we go to the one by one types in insect. So we have type 1 cells. So they have two common types of sensilla. Okay, so they have the first one is the trichoid sensilla, which they have the hair-like projections of sensilla. And the second common types of sensilla in the type 1 cells, okay, because of the sensilla structure, the cellular structure of the type 1 cells is a ciliated bipolar neurons. So they have two common sensilla. The first one, the trichoid, and the second one is the campaniform. Okay, campaniform is like bell shape. Okay, and the sensilla like this one. So this is the sensilla. So the sensilla is and arthropod sensory organs that protruding from the cuticles. Okay, protruding from the cuticles of the exoskeletons, or sometimes they are lying within or beneath the exoskeletons. Okay, so that is the sensilla. So the type 2 cells in the insects, so they have the scolopedia. Okay, the type 2 cells is internal surface. Okay, because the type 2 cells are associated with muscle or connective tissue. So they are internal surface. They are in the internal surface. So they have the scolopedia, which is bipolar neuron and complex accessory cells. Okay, so they have the scolopale here, and these type 2 cells can be isolated or grouped to form the chordotenal organ. And these type 2 cells are most function in proprioceptions, which is monitor the positions of the body of the insects. And these types 2 cells can be modified into tympanal organs. So tympanal organ is functioning in sound detections. Okay, so this is the function of type 2 cell in insects. So we move to the vertebrate tactile receptors. So these tactile receptors are widely dispersed and function as isolated sensory cells. They have a free nerve ending or enclosed in accessory structure. For example, they have a pacinian corpuscle, okay, in here. And they are functioning in pressure and vibrations, okay. So the tactile is actually related with the touch, okay, the touch. So proprioceptor. So the proprioceptor, as you know, is monitor the positions of the body. So we have three types of proprioceptors. The first one is the muscle spindles. Okay, here. So we also have the Golgi tendons organ. We also have the joint kinesthetic receptor. So the muscle spindles is to detect how much of a muscle is stretched. Okay, which is the muscle spindles are stretch receptor within the body of a muscle that primarily detect the changes in the length of the muscles. Okay, in the length of the muscle. So that is the function of the first type of proprioceptors, which is muscle spindles. We also have the Golgi tendons organs, which is detections of the tensions of the muscles on its tendon. Okay, here. And also to provide information about the strength and contractions and tensions of the muscles. Okay, so that is the second types of proprioceptors. And we have the third one is the joint kinesthetic receptor. So joint kinesthetic receptor here, so they are mechanoreceptors in the joint capsules, and they are detect They functioning in detect the angle and movement of the joint. Okay, movement or angle of the joints. So all the information will send to the our CNS, our central nerve systems. So equilibrium and hearing. Okay, so this is utilize the mechanoreceptors. So equilibriums, or we also said as a balance, it will detect the positions of body relative to the gravity. So this is equilibriums. We also have hearing. So hearing will detect and interpret the sound wave. So in vertebrate organisms, okay, the ear is responsible for both equilibriums, the balance, and also the hearings. While for the invertebrate animals, the organ for equilibriums are different from the organs of hearing. For example, the tympanal organs, okay, that is the organ for detecting the sound. So statosis. So statosis is an organ of equilibrium in the invertebrates. They are hollow and fluid-filled cavities that line with mechanosensory neurons. Okay, so it contains of the statolith. Statolith here, as you can see from here. So this is the statolith. Statolith is the dense particles of calcium carbonate. Okay, so this is the sensory neurons. Okay, so this is the statosis, which is the organ of equilibrium, the monitor of the body balance, okay, in invertebrate. So we also have the hair cells. Okay, just now is for equilibrium, so this is for the hearing. So mechanoreceptor cells used for hearing and balance in vertebrates, and it modified epithelial cells, and the hair cells that they have the extension extracellular structures and cilia that extend from the apical end. Okay, so this is the apical end, so they have the extracellular structure and cilia. So they have the stereocilia here, and also they have one kinociliums. So to join within the stereocilia and also kinocilium, they have a thick link. So this is the hair cells. Okay, so this is the hair cells, and this is the nucleus. So this is the afferent sensory neuron. So between these hair cells and also the afferent sensory neurons, we call it as a, what we call it? Sekejap, Huh? Sekejap. I'm sorry. Yeah. Okay. All right, sorry. So between these hair cells and also the sensory neuron, afferent sensory neuron, so this junction we call as a synapse. Okay, synapse gap. Okay, so the hair cells. So these hair cells can detect the movement and also the directions. So this is the signal transduction in hair cells. So at rest, so this is a at rest situations. So the hair cells slightly depolarize and release moderate amount of neurotransmitter. Okay, on the primary afferent cells. Okay, so this is the primary afferent cells. So they are releasing moderate amount of neurotransmitter in the rest situations, and also they will cause the intermediate frequency of AP. Okay, while when they are receive high amount of pressure signals, it will cause the stereocilia to pivot towards the kinociliums. Okay, so the mechanical gate will open. The mechanical gate will open, and additionals of K is the potassium. Additional of potassiums will enter the cells. Okay, as you can see from here. So it will cause the depolarizations, open voltage gated of calcium channels. Okay, so I will open, so the gate will open, so it will increase the and also this will increase the release of the neurotransmitter. So with the increasing of the neurotransmitter, so it will have increase of the frequency of the AP, the action potential here. So it create the depolarized situations. While in the C, so if they receive some sort of decreased light pressure signal, it cause the stereocilia to pivot away, okay, away from the kino cilium. So this is the kinociliums. So the mechanical gate here will close. So it will cause the hyperpolarizing. So the voltage gate calciums also will be closed. So it will decrease the release of the neurotransmitter and also will reduce the frequency of action potentials. Okay, so this is how the signal transductions in the hair cells. So in fish, so fish consisted the non-tetrapod vertebrates. Okay, so in the fish they use the hair cells in ears for hearing and for detecting the body positions and also orientations. So fish have the neuromas, okay, which neuromas can detect the water movement. So neuromas is actually the hair cells and also they also comprise of the accessory cupula, as you can see from this figure. So they have the cupula, which is filled with viscous gels and also hair cells. So this is we call as a neuromas. So the fish also have the lateral line systems. So an array of neuromas within pits or tubes running along the sides of the body. So these neuromas will detect the water movement in the fish. So the vertebrate ears. So vertebrate ears functioning in both equilibriums and hearing. So they have three part of ear, which is they have the inner ear, they also have the middle ear, and also they have the outer ear. So this is the pinna. Okay, the tunnel, the funnel that receive the sound. Okay. So in vertebrate ear, they are functioned in both for equilibrium, the balance of the body, and also for hearing. And for the equilibrium, the vestibular apparatus will detect the movement. Okay, so the vestibular apparatus is actually the three circular canals that comprise of the enlarged regions at the one end, which is we call as the ampulla, and also two sac-like swellings, or we call as the utricles and saccule. And all of these regions, they are content of the hair cells. Okay, so this is the examples of different organisms, different animals. We have the tailless fish, frog, and also they have birds. So and also mammals. So as you can see from here, so the tree is the circular canals. So they have the ampulla and also utricle and saccule, okay, in the tailless fish. So this is the structure of the semicircular canals in the frog. Okay, so this is the birds, and this is for the mammals. So they have the ampulla and also content of utricle and saccule. So all of different animals, they will have different structure of the vestibular apparatus, which is function in the text of the movement, okay, of the animals. So still in the vestibular apparatus, so if you remember, the vestibular apparatus is comprised of the two satellite swellings, which is utricles and saccules, and also the enlarged regions at the at one end we call as the ampulla. So in the ruticle and saccule, they are contained of mineralized otolith. Okay, so remember we're talking about the otolith just now, which is suspended in a macula covering more than 100,000 of hair cells. Okay, and while in the ampulla, there are lack of otolith, and also they are contained of cristae. The cristae is the hair cells that located in the inner cupula. So as you can see from here, so this is the macula of an utricles and saccules. Okay, so at the upper part of the gelatinous matrix, they have the otolith. Okay, so they also have the hair cells. So at the B, they have a crista of an ampulla. So they contain of the cristae, which is the cells that located in the cupula. So this is actually the cupula. Okay. So the function of ventricles in mammals. Okay. So so they have in ventricle they have a otolith here. Okay. So when the mammals are in the rest or constant motions, so they are actually partially depolarized. So but if the movements, if the mammals are move forward, so the hair cell will depolarize, which is they will release more neurotransmitter and result in they will have more frequency of action potentials. Okay. So functions of the cristae, which is detect the angular of accelerations. Okay. So as you can see from here, so this is the ampulla and also this is the cupula. Okay. This is the hair cells. All right. So this we call as a neuromass, which is the neuromass is function in the sense of the pressure. Okay. So this is the semicircular and also this is the ampulla. Okay. So at the rest situations, so the organ will partially depolarize. Okay, but when the animals are accelerations to the left, so they will have acceleration to the left. So this is the hip rotations movements. So they will have the polarizing, okay, and it will decrease the frequency of the AP, as you can see from this graph. And when the animals are accelerations to the right, okay, the head are rotate to the right, so they will have the depolarization, which is will increase will increase the frequency of the AP, okay, the action potentials. So we move to the sound detection. So inner ear, you remember ear in the vertebrate, they have three parts. They have outer ear, inner ear, and also middle ear. So the inner ear is actually to detect the sounds. So in fish, the incoming sound wave cause the otolith to move, which bend cilia or of the hair cells. So some fish will use the swim bladder to amplify the sound, to strengthen the sound. So this is the swim bladder. Okay. They use this swim bladder to amplify the sound. While in the terrestrial vertebrates, so the hearing will involve all of the parts of the ear, will involve the inner ear, middle ear, and also the outer ears. But the problem is the sound transfer between the air and also the fluid field in the ear are really poor. So what is the solution for this problem? So it will amplify the sounds. Okay, it will amplify the sound, it will strengthen the sound. So they have the pinna, if you remember the pinna. Okay, so the pinna is act as a funnel to collect more sounds. And they also have the middle ear bones. So the middle ear bones will increase the amplitude of vibrations from the tympanic membranes here, okay, to the oval window, okay, to the oval window. So the sound will enter here, so it will amplify, increase the amplitude of vibrations, okay, from this tympanic membrane to the oval window. So this is how they strengthen the sound in the terrestrial vertebrates. Okay, in the mammalian inner ear, so the inner ear in the mammalian are specialized for sound detections. So they have cochlea. Cochlea is a coil in mammals. So they also have the perilimb and endolimb. Okay. So the perilimb is fills vestibular and tympanic ducts. Okay, as you can see from this. So this is the tympanic ducts. Okay, and it's similar to the extracellular fluids. So it's similar as the extracellular fluids. While the endolimbs is filled with cochlear ducts. Okay, and is high in the potassium and also low in sodium. So organ of Corti here. So they have the contents of the hair cells and sit on the basilar membrane. Okay, so they have two types of cells. Yeah, they have the inner cells and also they have the outer cells. So the inner hair cells can detect the sounds, okay, functioning in detection of the sounds, while the outer hair cells functioning in amplify the sound. Okay. So the basilar membrane, okay. So the basilar membrane here, it is stiff and narrow at the proximal end, okay. It's stiff and narrow at the proximal end, and also flexible and wide at the distal end. So the frequency here, if they receive the high frequency, so the stiff end will vibrate. While if they receive the low frequency, so the flexible end will vibrate. Okay, so this is the sound encoding in the basilar membrane. So we move to the amplifications, the loudness. So the loud sounds will increase the movement of the basilar membrane, the vibrations of the basilar membranes, and also will increase the depolarizations of either inner hair. Okay, if you should remember, the inner hair is functioning in detection of these sounds, and also then it will increase the frequency of the action potentials. While the outer hair cells, okay, is functioning in amplify the sounds. So the outer hair cells will change shapes in response to sounds instead of releasing the neurotransmitter. Okay, and it will change the inner cell will release the neurotransmitter. Okay, and it will detect, it will make the epi frequency will be high, but the outer hair cells, so it actually normally will change the shapes in response to the sound instead of releasing the neurotransmitter. So this is the differentiation between the outer hair cells and inner hair cells. And the outer hair cells will change in shapes causes basilar membrane to move more and causes a large stimulus to the inner hair cells, which is to detect these sounds. So it will the outer hair cell will amplifies the sound. So this is the amplifications, okay, of the outer hair cells and inner hair cells. So the sound locations. So brain use informations on time lags, okay, on time lags and differences in the sound intensity. For example, if sound to the right ear first, if you hear the sounds to the right ear first, so it means that you can detect that the sounds are located to the right. But when the sound are louder, okay, if you hear some sound, if you hear sounds louder in your right ear, so it means that the sound are located to the right. Okay, so this is how the brains use the informations. Okay, which based on the lag, time lags also differences in the sound intensity. Okay, so we have done with the second type of stimuli. Okay. So we move to the photoreceptor, the third one. Okay, so photoreceptions is ability to detect a small proportions of a electromagnetic spectrum from ultraviolet to near infrared. Okay, so the concentrations on this range, okay, or wavelengths will support the idea that the animals evolved in water. Okay, which is it can detect a narrow band of it can detect, as you can see, it can detect a narrow band of electromagnetic spectrum in the visible light range. Okay, so this is the receptions, which is ability to detect a small proportions of the electromagnetic spectrums. Okay, so the organ range from single light sensitive cells to complex eyes, forming sharps and focus image. Okay, so they have two types of receptor. Okay, we have ciliary photoreceptor, and also we have the rhabdomeric photoreceptors. Okay, in the photoreceptor we have two types of receptor, ciliary and rhabdomeric. So for the ciliary photoreceptor, they have single highly folded ciliums, and also they have they fold from discs that contain of the photopigments. Okay, so the photopigment is actually the molecules that absorb the energy from the photons. Okay, so the second types of receptor in the photoreceptor is the rhabdomeric. So it actually have the apical surface. The apical surface is covered with multiple outfoldings we call as the microvillar projections. So this is the second type of photoreceptor. Okay, again, photopigments is molecule that absorb energy from photons. Okay, we focus on vertebrate. Okay, we focus on the vertebrate first. So the vertebrate photoreceptor, all photoreceptor of the vertebrates are ciliary photoreceptors. Okay, if you still remember the ciliary photoreceptor, they have a single highly full ciliums and they full from this and contain the photopigments. And for the ciliary photoreceptor in the vertebrates, okay, they have two types, which is they have rods and also they have cones. So this is the rod photoreceptor and this is cone photoreceptor. Okay, so this is the outer segment, this is the inner segment. Okay, so this is the body of these cells, and also this is the synaptic terminals. Okay, which release the neurotransmitter. So this is the characteristic of the rods and cones. Okay, so both have ciliary. Okay, the structure of the photoreceptor, they have both auxiliary. Okay, shapes. So the outer segment rod shape, the cones, the outer segment cone shape. So the sensitivity, this is the most important. Okay, the differentiation between rods and cones photoreceptor. The sensitivity for the rods, they are sensitive to very dim light. While for the cones, okay, the sensitivity for the cones photoreceptor, they are very sensitive to brighter light. So type of photopigment, okay, the molecules that absorb the energy from the photon, those they only have one type, while in the cone they have up to three types in mammals. So normally the nocturnal animals they will have relatively more roots, okay, because the roots are functioning in because the roots are sensitive to the very dim light. So we go to the photopigment. Okay, just now we're talking about the photopigments, the molecule that absorb the energy from the photon. So the photopigments have two covalently bonded parts. Okay, the first one they have chromophore. The second one they have opsin. Okay, the chromophore is actually the pigment that is derivative of vitamin A, for example, retina, and also the opsin is the protein, okay, the G protein couple receptor that involved in the steps, involved in the process of transduction in the photopigment. Okay, so this is the step in photoreception. So the chromophore will absorb the energy from the photon. So the chromophore will change the shapes. So the photoreceptor protein change the shape. So the protein used here is the opsin. Okay, and the signal will have the transduction cascade. Okay, later on we study about the signal transduction cascade, and the last step will change in membrane potentials. Okay. So that is the step in photoreceptions. So for the phototransductions, okay, it's actually the transduction cascade differ in rhabdomeric and ciliary photoreceptor. Okay, it different in both type of photoreceptor in the photoreception. Okay, so the first one, this is the phototransduction in the rhabdomeric photoreceptor, and the B1, the below figure is actually the process, the process. Okay, the steps that happen in the vertebrate photoreceptor. Okay, so for the step 1, step 2, so in here they also involve the G protein here, all right, and they will involve the channels which is TRP, and also they will have they will result in the depolarizations. But in the phototransition invertebrate photoreceptor, they will result in the hyperpolarizations, which is the sodiums, okay, will enter the cells. Okay, okay. So the eye. So the eye spot are single cells or regions of a cells that contain photosensitive pigment. Okay. So So they have, this is the structure. The eyes are very complex organs. Okay, they are very complex. So this is the structure of a major types of animals eyes. So the difference between each structure, we have the flat eye sheet, we have the vesicular eye, cup-shaped eye, and also convex eyes. So it's actually based on the arrangement of the photoreceptor cells. Okay, so this is the photoreceptor cells. Okay, so it are based on the arrangement of the photoreceptor cells. So we go one by one. The first one is the flat sheet eyes. It will provide some sense of light directions and intensity. So most often seen in larvae forms or as accessory eyes in adult. Okay, so this is the retina. Okay, so this is the photoreceptor cells. So it will receive the stimulus from here and will release the neurotransmitter. Okay, and we'll go to the primary afferent neurons. And the second structure of the eyes is the cup-shaped eyes. So the retina sheet is folded to form a narrow aperture. So better discrimination of light direction and intensity. So this is the functions. Okay, they have the better discrimination of light direction and intensity, and normally you can see in the nautilus. Okay, so this is the retina. So this is the photoreceptor cells. Okay, it will receive the photo pigment from here, and it will go through some process, some step here, and we release the neurotransmitter here. Okay, and it will transmit to the CNS. Okay, so we go to the third structure, which is vesicular eyes. So they use a lens in the aperture to improve clarity and intensity. So they have a lens here in the vesicular eyes. So the lens reflects lights and focuses it onto a single point on the retina. Okay, so this is the retina. It will reflect the light and focus it onto a single point on the retina. Okay, so this is the arrangement of the photoreceptor cells. So yeah, the vascular size eyes are present in most vertebrates. Okay. So the last structure of the eyes is the convex eyes. So the photoreceptors radiate outward, forming a convex retina. Okay, forming a convex retina. So it present in the annelids, molars, and also arthropods. Okay, compound eyes. So most complex compound eyes is actually the most complex of convex eyes. Okay, and can be found in the arthropods. Okay, it composed of the ommatidia. Okay, the compound eyes they composed of the ommatidia, and it will form image in two ways. Okay, the first one is the oppositions compound eyes. The second one is superposition compound eyes. So in the opposition compound eyes, the ommatidium will operate independently, and the afferent neurons, okay, will make interconnection to generate an image. Okay, you remember the afferent neuron. Okay, the afferent neuron will make interconnection to generate an image. And for the superposition compound eyes, so the ommatidium will work together to form an image of the retina. Okay, so the ommatidiums will work together with the afferent neurons in order to form an image on the retina. So we go to the vertebrate eyes. So it will form bright and focused image. So this is the part, okay, that you actually just need to know. Okay, we have the sclera. We have the cornea here, we have the choroid here, we also have the septum. Okay, septum is a layer of chloride of nocturnal animals that reflects the eyes. Okay, in here. So and also we have iris, we also have pupil lens, we also have ciliary body, we also have aqueous humor and vitreous humor. So this is all the parts in the vertebrate eyes. Okay, so the retina. So the retina is actually arranged into several layers. So all of this is actually a retina. It arranged into several layers. So they also have the rod and cones. Okay, rod and cones of receptor here. At the back and their tips are faced backward, okay, in here. So the axon of ganglion cells will join together. So this is the ganglion cells. The axon of the ganglion cells will join together to form the optic nerve. Okay, so this is the optic nerve. So this is actually the arrangement of the retina, okay, in the vertebrates. So this is the signal processing in the retina. So the rod and cones form different image. The rods, which is sensitive to the dim light, is principle of convergence. As many as 100 rod synapse with a single bipolar, and many bipolar cells will synapse with a ganglion cells. So it will produce a large visual field, but they will also will produce a fuzzy image. Okay, while for cones, okay, they are sensitive to the brighter light. So one cones will synapse with one bipolar cells which connects to one ganglion cells, and they will produce a small visual field but with high resolution image. Okay, so this is the signal processing in the retina. Okay, so this is the structure, okay, line in the retina. So the complex on and off regions of the receptive field of ganglion cells improve their ability to detect contrast between light and dark. Okay, so this is all the process involved. Okay, but in here, in the bipolar cells will depolarize, while in here the off region, the bipolar cells will hyperpolarize. So if the bipolar cells depolarize, so it will increase the releasing of the neurotransmitter. Okay, it will increase the releasing of the neurotransmitter here through the synapse. Okay, and if the bipolar cells are hyperpolarized, so it will decrease the releasing of the neurotransmitter here. Okay, and it will result in high response to light focus. Okay, and when there are hyperpolarizations, the light focus on the surrounding regions. Okay, so this is the on and off regions of receptive field. Okay, so how the brain processes the visual signal? Okay, so this is the signal, the photo pigment that receive. Okay, so then after just now, after the photo pigment had been processed here, so it will transfer the information to the nerve, to the optic nerve. Okay, so then I will transfer to the optic nerve, then we transfer the information to the optic chiasms here. So it will also continue the information will transmit through the optic tracts and lateral geniculate nucleus. Then last, okay, it will transmit to the visual cortex here. So each side of the brain, okay, it will receive the information from both eyes. Okay, so this is how the brain processes the visual signal. Okay, so we are done with the photoreceptors. Okay, so we move to the fourth type of stimuli. Okay, which is the magnetororeceptor. So this is very simple receptor. This is very simple type of stimuli receptor. So the magnetororeceptions, it is the ability to detect the magnetic fields. Okay, so magnetororeceptions, so the ability to detect magnetic fields. For example, the migratory birds, the homing salmon, we also have the butterfly. Okay, that normally the animals that apply the migrations. Okay, so the neurons in the olfactory epitheliums of rainbow trout contain a particle that resemble the magnetite. Okay, so this is all the magnetite here. So for example here, many mammals appear to use the earth magnetic field lines, which is to orient themselves as they migrate. So for example in here, the whales, apparently they sense earth magnetic field and use the information along with other cues for orientations. Okay. And also the other examples is the monarch butterfly. So this group of monarch butterfly, they use the magnetite, okay, to orient themselves with respect to Earth magnetic fields and navigate during the winter migrations. Okay. So that is the fourth types of stimuli. Okay, we move to the last classifications, okay, of the type of stimuli, which is thermoreceptors, which is regarding the temperature. Okay, so thermoreceptors are specialized to respond to the physical stress of temperatures, okay, either above or either below the animal's body temperature. But it is unclear whether these receptors are more like the mechanoreceptor, which is regarding the mechanic, okay, or operate by some of other mechanism. Okay, so for example, in here we have central thermoreceptor. So it located in the hypothalamus, which is in function to monitor the internal temperature. We also have the peripheral thermoreceptors, which is functioning in monitor the environmental temperature. Monitor the warm or cold, and also we have the other type of environmental temperature. We have thermononiceptors. So the thermononiceptors is functioning in detection the painfully on hot stimuli. So we have the thermo-TRPs. So this thermo-TRPs is actually the ion channel of thermoreceptor protein that involved in the process of thermoreceptors. Okay. So specialized thermoreceptions. So they have animals that have some animals, they have the specialized organs for detecting the heat radiating object at a distance. Okay, so for example, the specialized organ is the pit organs here. So the pit found between the eye and also the nostril of the pit vipers. So this pit organ can detect two 0.003 degrees Celsius changes, okay, and 0.5 degrees Celsius changes for humans. So for example, here some snakes have very sensitive infrared receptor, which they can detect body heat of the prey against a colder background. Okay, so this rattlesnake here, so this is the example of rattlesnakes, and also other pit vipers have a pair of infrared receptor, which is one between each eyes and also nostrils. So these pit organs, okay, are sensitive enough to detect the infrared radiation emitted by a warm mouth and a meter away. Okay, so the snake will move their heads from side to side until the radiation is detected accurately by the two receptors, and then it will indicate that the mouse is straight ahead. Okay, so I think we have done with our lecture in the topic of sensory system. Okay, if you guys have any questions regarding this topic, so you can WhatsApp me or email me. Okay, thank you very much. Assalamualaikum. Sampai ke mana? Shh, shh, shh. Jap, jap, jap.